Cysticercosis is a parasitic infection caused by the larval stage of the pork tapeworm Taenia solium. This condition occurs when the larvae, known as cystic ursi, invade various tissues and organs of the body. The most common sites of infection include the muscles, brain, and subcutaneous tissues. Humans can acquire cysticercosis by ingesting the eggs of the pork tapeworm, which are usually present in contaminated food or water. In this study on cysticercosis, a worldwide parasitic disease affecting humans and pigs, researchers utilized the recently available Taenia solium genome to identify a novel gene within a 1,448 base pair in open reading frames. The theoretical product of this gene is a 433 amino acid long alpha enolase, closely related to enolases found in other flatworms. Bioinformatic analyses further revealed a putative plasminogen binding region on this protein, suggesting a potential in pathogenesis. To explore its functional and diagnostic potential, the researchers isolated the mRNA encoding for this enolase from Taenia solium metacestodes, reverse transcribed it into chromosomal DNA, and subsequently cloned and expressed it in both E. coli as recombinant enolase from Taenia solium, or ReNOTs, and insect cells as recombinant enolase from Taenia solium and baculovirus, or ReNOTs-BAC, each with a 6' histidine tag. The molecular weights of these recombinant proteins were approximately 48 and 50 kilodaltons, with differences likely attributed to glycosylation variations. Spectrophotometric assays confirmed the enolase nature of ReNOTs, revealing enzymatic activity with specific activity at 60,000 units per milligram and a Michaelis constant at 0.091 millimolars, similar to enolases of other flatworms. Additionally, ReNOTs exhibited high immunogenicity, eliciting a robust polyclonal antibody response in immunized rabbits. The researchers leveraged ReNOTs-BAC for an ELISA aimed at discriminating between healthy and Taenia solium infected pigs. This diagnostic assay demonstrated a sensitivity of 88.4% and a specificity of 83.7%, showcasing its potential as an effective immunodiagnostic tool for porcine cysticercosis. In conclusion, the study not only unveiled and enzymatically characterized a novel enolase from Taenia solium metacestodes, but also presented a promising avenue for its application in disease diagnosis.
The methodology of the study on a novel enolase from the Taenia solium metacestodes and its potential as an immunodiagnostic antigen for porcine cysticercosis unfolds in a systematic and comprehensive fashion. The investigation commences with the alignment of the predicted amino acid sequence of T solium enolase against related platyhelminth sequences using the Clustal W. This initial step includes a separate alignment to assess conservation with enolases from common porcine helminths. Subsequently, the study delves into phylogenetic analysis, employing the maximum likelihood approach and a diverse set of enolase sequences from various organisms. The structural homology modeling utilizes Swiss model with a PDB crystal template, predicting active site residues and identifying key functional motifs through bioinformatic tools. The RNA extraction from the porcine cysts follows, utilizing the guanidinium thiocyanate phenylchlorophyll method, leading to reverse transcription into cDNA. The next phase involves cloning, expression, and purification of recombinant enolase, initially in E. coli and subsequently in a eukaryotic system using a baculovirus vector. The process includes the construction of a recombinant baculovirus shuttle vector and transfection into the SF9 insect cells, ultimately leading to the expression and purification of the ReNOTS-BAAC. Enzymatic activity assays elucidate the functionality of the recombinant enolase of the T solium, measuring the conversion of the 2-phosphoglycerate to phosphoenol pyruvate simultaneously. Rabbits are subcutaneously immunized with purified recombinant enolase of the T solium, and the ensuing antibody production is assessed through Western blots. The evaluation of the recombinant analyse of the T-Solium for immunodiagnosis involves testing porcine serum samples through ELISA immunoassays, utilizing the sensitivity, specificity, logistic regression, and the ROC analysis for data interpretation. Stringent ethical protocols underscore the study, with approvals from the Institutional Ethics Committee for Use of Animals of Universidad Peruana Cayetano Heredia. Samples are sourced responsibly from the Cysticercosis Working Group in Peru, ensuring a judicious approach to the study of annulase in the context of T-Solium cysticercosis. To summarize the methodology, the meticulous design encompasses diverse techniques ranging from molecular analysis to diagnostic evaluations, contributing to a nuanced understanding of annulase and the parasitic infection. The incorporation of robust bioinformatic tools, recombinant protein expression, and thorough immunological assessment adds depth and credibility to the exploration.
The study evaluates the analyse's immunodiagnostic properties, aiming to develop a reliable test for detecting cysticercosis in pigs. The research involved laboratory experiments and assessments of the analyse's sensitivity and specificity as an antigen for detecting antibodies related to porcine cysticercosis. The final objective is to aid in the creation of a diagnostic instrument that will be useful in the early identification of pig cysticercosis, which is essential for managing and stopping the spread of this parasitic illness.
In conclusion, the investigation into a novel analase from Taenia-Solium metastodes and its evaluation as immunodiagnostic antigen for porcine cysticercosis is rooted in the prevailing challenge of this parasitic disease. Cysticercosis continues to pose a significant global health threat, particularly in areas where pigs roam freely and sanitation is inadequate. With an estimated 50 million cases and 50,000 deaths annually worldwide, the burden of cysticercosis highlights the critical necessities for innovative diagnostic tools. The study has successfully expressed a functional recombinant enolase from T. solium in both prokaryote and eukaryote expression systems, effectively demonstrating its glycolytic activity. Moreover, the research underscores that recombinant enolase, particularly when expressed in SF9 insect cells, stands out as a promising candidate for future development of diagnostic tests for porcine cysticercosis. Overall, in the broader context of cysticercosis, the findings not only contribute to advancing diagnostic strategies, but also open new avenues providing fresh perspective for more precise and accessible detection methods. As we conclude, the significance of this enolase as potential diagnostic tools becomes increasingly apparent, making substantial contributions to the field of parasitology.